Hello everyone and welcome to my first video, and we're going to be investigating how to use restriction digests to try and diagnose genetic mutations. So for this we've chosen the example of sickle cell anemia, which is quite a well-known disease now. So with sickle cell anemia, we'll give a little bit of background, and as I say, I don't want to go too much into the theory and the background. You can find that in textbooks, other websites, plenty of other people on YouTube doing videos like these, but I just want to really get into the actual application of restriction digests and how we can use them to diagnose a mutation. But we'll give a bit of background first. So within a red blood cell, they're full of hemoglobin, and hemoglobin has this quaternary structure. And here you can see the alpha chains here and here, and then the two beta chains as well, there and there, each of which has their own heme group within it, and they transition between a relaxed and a tense state in their normal form, and this allows them to pick up and drop off oxygen and carbon dioxide across the body as and where needed. So what happens in sickle cell anemia is we get a single nucleotide polymorphism, and that is, as it suggests in the name, a single nucleotide that has changed its form. So this single nucleotide change that you can see here and here changes the nature of the whole triplet codon, which in turn changes the amino acid that it codes for. So here you can see the change in the amino acid sequence, going from a glutamine initially in a normal beta globin to valine in the mutant beta globin. And this addition of the valine causes a new patch of hydrophobicity. So here you can see where the valine 6 has been substituted in an example hemoglobin molecule of sickle cell anemia. You see this agglutination because the hydrophobicity is attracted to another patch of hydrophobicity. They both essentially want to get out of an aqueous environment. So you end up getting this agglutination of hemoglobin molecules, and this continues on and on and on and on and on until the end result is your erythrocytes have this sort of sickle shape, and hence the name sickle cell anemia. So how were we going to analyze this? Well, the first thing I'm going to do is introduce to you the bioinformatics package that I quite like using. So this is Benchling. Now, if you haven't got it already, I would highly recommend getting it, especially if you're in the life sciences doing a degree, or especially if you're going any further, if you're looking to do molecular biology, cloning, etc., etc. It's a really simple system to set up and to get used to, and it all keeps it online. It's browser based, there's no download, there's no hidden costs at all. So I'm going to jump in and sign into the Benchling account, which I've made previously. And here we are, and I've made a new account for this as well, so we've got a nice blank starting point. So the first thing we want to do if we want to analyze this gene is to find the gene itself. So for that we're going to need a genome browser. So here is one I already have open. Now I'm using this one only because I've used it before and I'm more familiar with it, but you don't necessarily have to use this. There are all sorts of genome browsers across the web, and it also depends what organism you're looking at. For example, if you're working with plants, in particular Arabidopsis thaliana, for example, which is a common plant organism, you'll use a site like TER, and you can find sequences all across in numerous types of databases, but we're just going to stick with this one for now. So what I want to do is go to the tools, and here there should be a gene sorter. There we go. So I'm going to click there. And then what we're looking for is the human beta globin beta subunit. So we're going to type in HBB and hit enter. And then we lined it in this list. There we go. There we go. And now we want to make sure that we output this sequence. So we're going to hit output the sequence. What type of sequence? So we want the full genomic sequence, okay? So we don't just want the mRNA or the protein sequence, but we want the actual genomic sequence. So make sure this little one here is ticked and click there. So now we're going to take a genome sequence, and do we want any other region? So I'm going to take about 600 bases upstream as its default, 50 bases downstream, which again is default, and leave the exons in uppercase. So this helps us identify the differences between exons and introns throughout the sequence. So hit get sequence, and now we have that, we can actually port that into our Benchling program. So I'm going to show you how quickly now. So here we go down to the plus to create something new. We want to create a new DNA sequence, and there we go, new DNA sequence. Now this is going to be linear because it's not a plasmid DNA, it's not a ring of DNA, it's a single line. And we're going to call this the wild type, so WT HBB DNA, just so we know what it is. So we click create there, and here we have this new entry. So you have a tab system in Benchling, so they'll slowly rack up along here, as you'll see as we go through. And I'm just going to copy all of this. Make sure not to take any of the name at the top. Then use Control C on the keyboard, click on here, and then Control V. So that's copy and paste, and there we go. Now, the first thing I'm going to do, which I'd recommend you do as well, because it might make this seem a little more sense as we go through, but I'm going to turn off all of the enzymes. So just hit none. So we just got our DNA at the moment, and we'll go through it bit by bit. Now, as you can see, you've got lowercase letters here, and then you've got uppercase letters here. So as we go through, you've got introns, exons, and an untranslated region, or UTR. So I'm just going to annotate all of these so we can find our way through the sequence a little bit easier. So I'm going to start highlighting all of this, and to create an annotation, you simply right-click, create annotation, and then this one I'm just going to call UTR, so untranslated region. So I'm going to change this to a red type color. There we go, nice. And hit save. So now we can see that that's the UTR. Then moving on, this is the first exon that we've come across, and the exon is only capital letters, okay? So exons are expressed and introns are not. So that's a nice and easy way to remember it in terms of protein formation. So I'm then going to create an annotation here, so we know that this is an exon. And I'm going to change this to a sort of beigey color, that'll do. Okie dokie. And then between the exons you have your introns. So I'm just going to keep on going. Let's move that out of the way. There we go. And I'm going to create an annotation for this. And this is going to be an intron. And we'll give this purple. There we go. Okay, so I'm going to quickly pause the recording while I finish annotating the rest of the sequence, but it only takes a couple of seconds to do. And we're back. So now you can see that I've fully annotated the sequence as we go through. So you've got the untranslated regions capping either end of the medley of exons and introns. So as I say, it's literally just following the uppercase and lowercase letters as you go through the sequence. Now, what we want to find is where the mutation occurs within the sequence. So we want our point of interest, and then we can highlight this. So we know that in normal beta globin we should have this DNA sequence of CCT, GAGG, and this is around where the mutation occurs. So we're going to see if we can find that in benchlim. So I'm going to move this to one side again, and then clicking on the benchlim window, use Control and F, so this will help you find anything, and just type CCT, GAGG. And here we can see that we found a result which is in exon 1, so this little area here. So now if I click on that, it highlights that part of the sequence, and then right click, and we'll create an annotation here, and this will be our mutation site. So I'm just going to call it mute site here and give it a nice luminous color so we can easily identify it. There we go. So now we know that this occurs right here. So what I also want to do now is create a mutant sequence so that we can compare the results that we get as we go through to our wild type. So this is our wild type DNA as it is. So if I then go into our projects folder, so up here, so this is where your projects are being saved, and you should have this entry here. So this is the one that we've created here. So now what I want to do is I just want to copy that. So I'm going to copy 2, and that's where it's going. And there we go. Now we have 2 copies. So now I'm going to rename this one, and I'm going to call this the mutant HBB DNA, because now what we can do with the assistance of our image here, we know that we need to swap the A between the GAGG for a T. So we know where our point of interest is, so we can do that relatively easily. So first I'm going to close this little nav box because we don't need that anymore because we've got it permanently annotated. And then you're going to open the mutant HBB DNA sequence. Make sure you definitely do it in the mutant sequence because otherwise this is going to get confusing. So now we want to find the CCT-GAGG, and this is the bit we want to swap for a T. So we just highlight that single base, right click, and you delete the base, and then making sure it goes between the GGG, we're going to insert a new base and we're going to put a T. Press enter, and then what you should have on this site now is CCT-GTGG. So this is what happens inside the mutant. Now going back to the wild type, we want to design some primers to go around this first exon since we know that's where the mutation occurs. So in order to do that, I'm going to highlight a region that's going to completely cover the first exon, but we don't want to go over the top. We don't want a size that's too big to work with PCR. So I'm going to highlight from 400 bases down to 900. There we go. And this, as you can see, has the untranslated region in it as well, so we've got a bit of room to work with. And then we also have the first exon in there in its entirety, and then the intron as well. So to design primers in this package, you just right click and then hit Run Primer Free. Now this used to be a standalone program quite a while ago, but it's been integrated into Benchling, which is super useful. So because of the selection we have on the sequence map, here we go back to the primer wizard and we use that selection, so from 400 to 900 bases. And then normally this list will be closed, but as I say, is you want to set this for a minimum size of 600 and a maximum of 700. And then we can generate primers up here. So we just click the button and it will find the best products for you. Now the one I'm going to choose is this one here, so the product of 631 base pairs. That looks pretty good. So all I'm going to do is click on this little box here which highlights these two sets, and then click Save Selected Primers. And then we're going to drop that into our same folder as the rest of our sequences as well. Hit Create. And now you'll have your primers designed and stored in there for you. So these should come up actually. Let's see if we can get them to come up again. Aha, there we go. So this here, so the forward one, that's your forward primer and your reverse primer. And in here as well, each one that you click on, you can see the individual ratio of GC, the melting temperatures, and the actual bases itself, if you want to check any of the parameters or the properties of the primers themselves. And because we've designed them here, it's nicely already attached them to our sequence. So we can see this here and here. So that's wicked. But now we also want to add these to the mutant DNA sequence. So this is a bit easier. We don't have to redesign them. You just go to primers, which is this little section over here, and attach existing because we've already created them. And then click Find Binding Sites. And what we get is these same primers that we designed a second ago that have both come up together. So we're going to attach these. And then when we close the Find Binding Sites tab, now we can see our primers are here on both the wild type and the mutant sequences. So now what we want to do is actually do the PCR. So we want to do the PCR in silico, or they call it that because it's in a computer, as a computer is made out of silicon chipboard, so in silico, hence the name. So to create a PCR fragment on here, and we're going to start with a wild type, so make sure you've got that selected. So to create a PCR product here is we need to link these primers as a pair, which has already been done here because we created them on this strand. So now that you've got these pairs, you just click here, and there you can see your product size is 631 base pairs. So we're going to create our PCR product. So you just click there, and then copy, and then again store in the same folder. And this is our product. So I'm going to rename this. So this is a wild type HBB DNA PCR product. Okay, here we go. And you can see as well as it's got our entire exon 1 in it that it's already annotated for us, and our point of interest. And then I'm just going to do the same thing again with the mutant. So I'm going to close this for a second, and we don't need the wild type DNA anymore, so I'm also going to close that. And here we're going to do the PCR again. So if you click here, you can see there's no link primers because we didn't design them on this one, we designed it on the wild type. So to make these primers a pair is nice and simple, is you click on the first primer and then you use the shift button on your keyboard. So you have to hold this down. So hold down shift and then select the next primer. When that happens, this little box will go green, so you can link the primers. So this is telling the computer that these primers work as a pair. Between this forward and between this reverse, this is the fragment that we want to produce. So again, we create a PCR product, copy, okay, there we go. And then I'm just going to rename this again. So we've got the mute HBB DNA PCR product. So that's us done for our in silico PCR. So now imagine we've taken the DNA from a wild type person and a mutant. And we've used PCR with these primers that we've designed, and they've bulked up this size fragment for us. So we've got a medley of samples. Now, what we want to do is see whether or not these people carry this mutation, and this is where it all starts coming together. So if we take the PCR product from the wild type, here we go, and now we want to use a restriction enzyme. So by doing the digest, we can actually see whether the mutation has occurred or not. So what we're going to do is click on here, and this will give us the digest option. Now, just to show you how often this happens, is I'm going to click all enzymes. And you can see just in this small section of the sequence alone, there are restriction enzyme sites everywhere across your own genome. And we can basically use these to highlight certain areas. So if you click on any given enzyme, it will show you where it cuts and what its recognition site is. But we don't want to go through all of these anyway. What we want to do is we want to find someone that cuts around our mutation site. So if we have a look here, you can keep on going through the whole set of enzymes. But here, now that is in a lovely position right there. So we have this BSU 361 enzyme that cuts right on top of the mutation site. So that's the one that we want to choose. So we're going to filter out all of these others. And what we're going to do is go for BSU 361. So this one's here because it recognizes there's one cut already. So we're just going to click that. If you can't find it, then you can search it nice and easy by typing in BSU 36. And there we go. So I'm going to click on that enzyme. So that one's been highlighted. And then we're going to click none here as well to sort of get rid of all of the others. Let's clean that up a bit. Okay, so now you can see where the enzyme actually cuts. So these arrows indicate where the position of the cut actually takes place. So as you can see, when you just hover over the top, is the CC and T is where the cut site actually occurs. And it happens the same as well on the opposing strand. So on the 5' to 3' and 3' to 5', it will cut in the same place depending on what the actual recognition site is. So now we've got this selected, and we can do an in silico digest as well. So if we run the digest here, this is our result output. Now, it looks a bit meddly at first, and you're thinking, oh, it's only cut once, why is there two sequences? Well, that's fine. If you get a bit of string and then cut it once, you end up with two bits of string that are different sizes. And this is essentially what it's letting you know, is the start to the end of the first fragment is from 1 to 490 with a total size of 490 base pairs. And then the second fragment is from 491 to 631, which is a length of 141 base pairs, which is pretty cool. So if you use virtual digest up here, what this will do is give you an idea of what you're expecting to see on a gel. So now we know that by using our primers on a wild type person aiming at this HBB gene, our PCR product, when it's cut with this BSU 361 enzyme, will give us two bands. So this is the letter here, and you can see the first band is about 500 base pairs, and the second is between 100 and 200 base pairs. So these are just your individual fragments that you've selected. So you can choose that there, and it will say 141 base pairs for your highlighted fragment here, and same again here. So this is just what you would imagine to see on a gel. So when you run your gel results on a agarose gel, this is the sort of output you're going to get, and that's the sort of size that you're after. So this is cool. We get one cut in a wild type, and we know now we get two bands on a gel of this size. So if you get two bands on a gel of this size from this PCR product, then you know that this person is not carrying the mutation for sickle cell anemia. Now, to contrast this, we're just going to do exactly the same thing again with the mutant PCR product, which is up here. So if we click here, we can do the same again. So I'm going to go onto there, choose a BSU 361, which is already selected, and then run digest. Now you see here we've only got one entry, so from 1 to 631, which is the whole PCR product size, so it hasn't actually cut. And when you look at the virtual digest, this is the one of the wild type, and this is the one of the wild type again. So this is after it's been cut once with the BSU 361 enzyme. But here you can see our second result. Now this second result is the total size of the PCR product. It hasn't been able to cut, and the reason why the enzyme cannot cut inside the mutant is because it doesn't have the CCTGAGG. It has these CCTGTGG instead, which means that the enzyme cannot cut. So when it comes to looking at your actual results, that's how you can compare and contrast whether or not you've got a wild type or mutant person. And that is pretty much it. So I hope that this has been useful for you. I'd really appreciate any feedback you give. I will apologise, the video's not completely perfect, or it hasn't had a massive amount of work done to it. This is my first one that I've done, but as I say, I really would appreciate your feedback. If you find this useful, and if there's any other particular subjects that you'd like to look at, or if there's anything else that I might potentially be able to help with, if I can't do the video myself, I'm pretty sure I could forward you onto some sites that have some wicked information as well. So cheers guys, and I hope it all goes well.